Swayam Prabha. Digital India. Educated India.
In this lecture, we will discuss on pairwise alignment. So, in the earlier classes, we dealt with different types of sequences, right, for the biological macromolecules. What are the different databases we use to obtain the sequences? For example, for the DNA sequences, EMBL, GenBank and DDBJ, right, DNA Data Bank of Japan. For the case of protein sequences, PAR and UniProt, right. So, if you have the sequences from different organisms, how far they are similar, how far they are different. So, the sequences whether they resemble each other and for any particular function. In this case, we need to compare the sequences and you have to align properly, and you can see the similarities and differences. For this case, okay, there are various techniques to compare the sequences, and we will discuss the details in this lecture. So, let us just refresh ourselves regarding the contents we discussed in the previous class. So, what did we discuss in the last class? Protein primary structure. What is the primary structure? Linear sequence of the protein, right. So, here the main chain is the same and the side chain is different, right. So, if you see, you can see NH2, C alpha, C, N, C alpha, C OH, right. So, here you have the hydrogen and the R group. So, main chain is the same, the repetition, right, with the elimination of water molecule, and here you have the side chain R1 R2. So, there are 20 different types of amino acid residues, right. You can form any chains, right, based on this R1 and R2. Then we discussed that nature selects particular specific combination of amino acid residues to form a functional protein. Then we discussed where shall we get the information. So, there are several proteins which contain the sequences. Currently, more than 70 million sequences are known, right. Where shall we get the information? So, initially we started to collect manually and publish a book called Atlas of Protein Sequences, right. So, then they developed this into the protein information resource called PIR in Georgetown University. In the meantime, the Munich Swiss Institute of Bioinformatics, right, they also tried to collect the sequences, right, for example, the SwissProt, and both the places they developed several tools to analyze the sequences. Later on, they formed a consortium, right, they made the UniProt. There is now universally accepted at the protein sequence database. Then what are the various features of UniProt? What are the major aspects of UniProt? A high level of annotation, minimum redundancy, and large high integration with other databases, right. So, it has various features in UniProt. So, they classify into three different groups. The first part you can see is general information regarding the sequence, regarding the structure and the function, and general information regarding a particular protein. And second part, we give the sequence information and structure information and interaction informations along with the links to other databases, right. And the last part they mentioned about the enzymes and pathways and so on. They have high level of integration as well as the links with all the other databases in the literature. Then how to obtain the data from UniProt? For example, if you want to obtain the data for transcription factors, how to get the data? Go to UniProt, search option, use transcription factors, then you will get a list of sequences. If you want to remove redundancy, you can also do the redundancy, but there are only few options available in UniProt. What are the various options available in the UniProt? 90%, 50% and 100%, right. You get all the sequences, right, 90% and 50%. So, if you want 50%, you select 50% and you can download the data, right, then you can do it for analysis. Then if you are interested on any specific protein, then you give that particular protein. So, you get the information regarding that particular protein. For example, last class we discussed about hemoglobin. So, we got the sequence, we got the function, you got the post translational modification sites and the binding sites, all the information you can get from UniProt. Now, we discuss about the alignment. What do you think about alignment? Yeah, so you can see how they evolutionally related, right. They evolved for very period of time. So, if you have one sequence and we have another sequence, right. You can see the one sequence here, and we have another sequence. How they are related with each other, how long it took to get different types of sequences. If you take the sequence 1 and sequence 2, you can see some differences. Here there is no difference, here there is no difference, and you can see a difference here. And then you can see the relationship between two different sequences to understand how they are similar, how are the different functions, whether they are affected by any functions, and so on. So, here I give two examples. Okay, first one is the human hemoglobin, and the second one is chicken hemoglobin. So, we look into these two sequences. Can you see any difference or similarities between the two sequences? Okay, here this is similar, and you can see some places, right, W, G, K, V, N, V, right. So, here you can see, right. So, when you look at your eyes, you can see some cases, but we require an algorithm, right, to quantitate how far these two sequences are similar to each other, which part of the sequences are similar, which part of these sequences are different, right. So, here again you can see K, Y, H is the same, but again here this is difference. So, in this case there are various methods available to compare these two sequences, right. This is called the pairwise comparison. The simplest one is dot plot, right. This will compare these two sequences, and if the sequences are the same, then we put a dot, right. I will explain in a couple of minutes, right. And then from this plot we can see whether the same residue is located at the same place, or there any shift, like this is one residue shift in the first sequence, or any shift in the second sequence, that they will see. So, this plot will give some information regarding, you can see some repeats if you have any region which is the same in between the two sequences, and you can see any long insertions or the deletions, for example, there is shift in the first sequence or the shift in the second sequence, and also you can see some portion of the sequence which can be aligned together for the different sequences. This is the first one. Then the second one we can do local alignments. Here you can see instead of the whole sequences, you can do it for some small portions, whether any small portions we can have the alignment between these two sequences. Then we do a global alignment will give you the whole alignment for the complete set of sequence. For example, if you see here, you can align for the whole sequence, or you can align only some specific portions of the sequence. How to align the sequences, how to score the sequences, that we will discuss in a couple of classes. So, what is the dot plot? What information you can get from dot plot? It is one of the simplest methods, because you have the sequence A and sequence B. So, compare these two sequences and make a dot if they are similar to each other, and you can get the similarity plot between the two sequences. How to do it? So, I will give you the one sequence here, the second sequence, right. So, compare the sequences, take sequence 1 and sequence 2, right. Here this is the x axis and here is y axis, this is amino acid sequence. Here put the amino acid sequence. So, if you have two sequences, I will write it clear. So, for example, I put the sequence here, right. So, I put another sequence here, so this is sequence 1, this is sequence 2. So, you can make a plot, right. So, do it here A, I, K, T, L, V, A, T, A, V, right. Go to the y axis. So, here is second sequence A, I, K, L, V, A, I, T, A, right. So, in this A here, so where are the A's present in the y axis? Here we have A, so then over here we have A, here we have A, here we have A. So, in instance I, where are the I's? Here and here one I, right. Then K, so K is here, that is all. Then T, okay, here you have one T here, right, here, here that is another one. Then L, one L is here, no other L's, right. Then V, where is V? V is here, then A again here, there is one A here, another A here, another A here, right. Then this A the same here, 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 then T here, this A here, 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 then V, one V is here, only one, right. So, if you see this one, so if you see these are the cases you can see the continuous dots. So, to put a line here, this continuous, and here this is a sound shift, and then you can see continuous line here, right. So, this will tell you if it is the diagonal, then what can we infer from this diagonal ones? Yeah. So, these three are diagonal, A, K same. So, here you can see this is the same, right, the position is the same. Now, second one, if you see, look at here, here this is a shift. So, you can see a shift between sequence 1 and the sequence 2. So, you can see, you can still see here LV AA here, you can see the same one here with a shift of one residue. So, if you have the sequences, it is difficult to see where are the residues which are exactly matched and where they have a shift, but if you make a dot plot like this is the dot plot, easily you can locate the residues where they matches and where they have the shift either in the first sequence or in the second sequence. So, here there is a shift in the first sequence because it is shifted here, right. So, you can say down. So, likewise if in the next another sequence you can have a shift at the above the diagonal, right. You can from this one you can see where you have the shift in the first sequence or the second sequence. So, what is the application of this dot plot?
So, we can find the alignment and also we can see if there is any shift from the alignment.
Right, you can easily see if you make a plot, immediately you can see that whether there is an exact match either at the diagonal or at the any of the up the diagonal or the down the diagonal. So, here this is how to construct the dot plots. First dots are placed in the space of each position where the sequence elements are identical, and then it will give you the identity between the two sequences, right, and the diagonals of this dot plot. So, this here we exactly looked at the same one. Then we can also use a different window size. You can use different window size and you can use some threshold. For example, if you take the window size of 5, so take this 5, compare this 5, and among the 5 how many of them are matching. So, then you put a threshold, for example 3. So, you have 3 matching 1, 2, 3, then you can say okay there is a match. Then go to the next window, this 5, then whether put the threshold of 3 if it matches or not, if it matches put a dot. Then you can get different types of plot. If there is a one sequence difference, then you will consider that specific difference because they make the window average, right. Either you can 3 or 4 you can put, right. So, you can get a plot, right, with a smooth plot with respect to the two sequences, sequence 1 and sequence 2. With a single mutation, right, here we do not find the difference, but if we make a real dot plot with the same sequence, then you can see a difference. Even a mutation you can see a difference, right, that is a difference. Okay. So, this is a protein data. So, here x axis is the amino acid sequence, y axis amino acid sequence, right. Here we have about 150 amino acid residues, right. So, you can see some regions which are similar, right, this one, right, we have the sequence. So, if we see there are some regions which are same, first few residues are same, then again some residues are same. So, you can see the regions where you have the long stretch of residues, that is around 100 residues. In the sequence, if you have the 100 residues, you can see the difference of the many residues, they have the closely related with each other. So, now we can for any proteins you can make a plot, right, and you can analyze where are the regions where they are similar to each other between any two sequences. So, then what is simple alignment? The simple alignment that I discussed earlier, just here that sequence 1 and here sequence 2, right, just how far they are aligned with each other, that means also how about the match between the characters in each sequence. So, I will show if you have different sequences, for example, you put 2 sequences, and you put another sequence here, For example, if you have this one AIT VSA and AIT AA. So, you can make these two sequences align with each other with the different aspects. One, that is a replacement of characters. For example, if you see here, so this here valine is changed to alanine, and this type of changes is called mutation or substitution. So, if you change or replace one amino acid by another amino acid, and that is called mutation. There are two other aspects, one is called the insertion. Here we add more data, more sequences, right. For example, if there is AIT, if you add any of the amino acid in between from anywhere, for example, we add S, then this will become AIST, this is called the addition. So, first one is the mutation. Mutation refers to the change of amino acids, right. For example, if there is a and with this is changed to V, or it is V or changed to A, this is mutation. Insertion, that adds one residue, right, in any sequence. Then there is another called deletion. In this case, some residues are deleted in the sequence. For example, if there is a sequence AIST and this I is deleted, then we get the sequence AIST, right. Here this I is deleted. So, these are the three kinds of changes in the sequences, either you have mutation or you have the insertion or you have the deletion. Then if you look into these sequences, the insertion deletions, they occur less frequently than mutations. If you see different sets of sequences from the same organisms, you frequently see the mutations, changing one amino acid by another amino acid, right. So, but less possibility of insertion deletions, but the insertion deletions we can represent by gaps, right, but this is not frequent. So, this will influence with the high penalty when you make a sequence alignment. Okay, now I showed two examples. First, this is the sequence 1, here sequence 2. So, how far these two sequences are related with each other? So, first, as we discussed, we can have dot plot, easily we can see. So, you can construct dot plot and you can see where we are you can have the same amino acids or in insertion deletions. Then we need a numeric system to evaluate how far these two sequences align with each other. They align well, or this is a good alignment or a bad alignment. So, we need a score. In this case, we need to develop some of these matrices to score whether sequence 1 is related with the sequence 2 or not. So, in this case, we need the scoring measures. So, we need 3 different types of measures we need, right, because we discussed about 3 different types of changes. What are 3 different types of changes? Mutation, insertion and deletion, right. So, insertion and deletions we represent the gap. So, we give the gap penalty. For the mutations, right, we need to give the penalty depending upon the types of mutations. Then some cases we will have same residue, there is no change. So, that will be rewarded. So, because that maintains the same residue. So, the two sequences are similar to each other.